In previous lecture, we saw the introduction about electron microscope, and we just saw two types of electron microscope, that is transmission electron microscope and scanning electron microscope. So in this lecture, we will study in detail about transmission electron microscope and uh, other type, that is scanning electron microscope. So coming to transmission electron microscope, it is a microscopy technique in which beam of electron is transmitted through the ultra thin specimen, interacting with the specimen as it passes through it. A transmission of electron microscope can be achieved better than 50 picometer resolution, and magnification is up to 10 lakhs x, means we can magnify to that much time. So we will study in detail about transmission electron microscope. So this is the structure of transmission electron microscope. It basically consists of high voltage electron gun. So electron gun is placed on the top of the transmission electron microscope, and it is act as a source of illumination. So just like we use light in light microscope, we use electron gun to deliver this electron as a source of illumination. First, condenser lens, means the electron passes through first condenser lens, then it passes through condenser aperture, then it passes through second condenser lens, and then condenser aperture, specimen holder, air lock, objective lens and aperture, electron beam and fluorescent screen and camera. So what they basically do, they keep their specimen, means a thin section of a specimen is placed on the objective lens, that is specimen holder, and they made very thin section of that specimen. Why they made very thin section of specimen? Because they this electron should pass through that specimen, so the section should be very thin. If the section is a little broader, then the electron cannot pass through that beam, and you will not get a proper image. So while designing or while preparing your specimen, it should be very small, and section should be, means just like you use the stem of plant to take section. So in this technique, you also have to take very thin section so that electron can pass through it and you will get a proper image. And finally, when electron pass through that specimen, at the bottom of the electron microscope there are fluorescent screen and camera. So whatever the image, means electron pass through that some specimen, and it cast an image on the fluorescent screen, and that image you can see on your laptop or whatever computer connected with that electron microscope, and you will get a very beautiful and a sharp image by using this transmission electron microscope. Now coming to different types of transmission electron microscope, that is, just you have to remember the name, you don't have to go in detail about this types of transmission electron microscope, like bright field transmission electron microscope, dark field transmission electron microscope, high resolution transmission electron microscope, energy filtered transmission electron microscope, and electron diffraction transmission electron microscope. So these are the different names given to that transmission electron microscope. You don't have to go in detail and study each and every time, you just have to remember the name of each of the transmission electron microscope. Now coming to its application and limitation of transmission electron microscope. So we will see the application one by one. That it can be used to study of to examine the small column of the atom, means in a specimen. It can even focus a particular atom, and it can examine the structure of that atom. Cancer research, this is widely used in cancer research, as cancer is already taught. They are basically continuous dividing cells, so we can take a sample of that cancerous tissue and or cancerous cell, and we can see its structure by using transmission electron microscope. Virology, you must be very aware at this point of time, because everyone is talking about corona. So we can see the minute structure, means every crown, every detail about that particular virus by using this transmission electron microscope, which is impossible by using simple microscope or light microscope. We can't see virus because they are very small, but by using transmission electron microscope we can see the minute detail about transmission virus. So it is used in virology. Pollution nanotechnology means we know that different particles, means airborne particle, cause different type of pollution. So we can observe different types of particle which can cause pollution in the environment by using transmission electron microscope. Semiconductor research, you must have used semiconductor in your 12th by in your physics lab. So to develop that semiconductor, we uh, we take use of microscope. Mm, that is transmission electron microscope. Chemical identity means if you take a particular chemical like calcium carbonate, so we can differentiate calcium and carbonate by using this transmission electron microscope, and we see the particular structure about any chemical, means we can see the minute atomic structure by using this transmission electron microscope, and uh, we can identify that chemical by looking on it. Next is crystal orientation. There are different type of crystal you will study on in your further classes, like uh, quartz and other crystals. So it is very minute, so we cannot see by naked eye. So by using this transmission electron microscope we can observe crystal structure, and they are so beautiful, structurally organized, that when you see that crystal you will amazed about it. Next is electronic structure, means even um, any electronic structure which is impossible to observe by naked eye, we can make use of transmission electron microscope to observe this electronic structure. These were the advantages of using transmission electron microscope, but there are some limitation means some disadvantages of using this transmission electron microscope. So we will go through each of these. It is difficult to produce thin sample, as this technique require very thin sampling so that electron can pass through it. This thin sampling is very difficult to make. We should make that much thin that only a single layer remain, and that is means it is a very tough task to make that much thin section. Next, relatively time consuming process with low throughput or sample. And if we are using this transmission electron microscope, then it is a very time consuming process. Means if we are using simple microscope, we just have to take our sample, and we have to focus by using light microscope. But if we are using this transmission electron microscope, this is very time consuming. First you have to make different section, then you have to arrange, and installation cost is very high, so you have to invest a lot in this electron microscope. So these are the some disadvantages. The structure of sample may be changed during preparation process, and we are observing this sample. So while preparing sample, we don't use live organism. The specimen which we use for transmission electron microscope um, must not be live, because we take a thin section, and when we are preparing this thin section, the structure might change. So when we observe that structure, so it's possibility that we will not observe actual structure of that specimen, because while preparing, the structure might get changed, and that change structure we will observe after transmission. Next is small field view may not give conclusion result of the whole specimen. And this transmission electron microscope, we can focus on a very small area. So by focusing on very small area, we will not get idea about whole specimen. Mm, just like imagine we are using simple microscope and we are observing the structure of chloroplast or anything, so we can observe entire structure. But if we are using this transmission electron microscope, it will focus on very minute area. So by focusing on that minute area, we will not able to observe the entire specimen, so we will get idea about only some part. So this is one of the limitation. And this was the transmission electron microscope. In next lecture we will see more about the uh, scanning electron microscope. Thank you.